Welcome to the 6th edition of the International Dairy Federation Nutritional Health Symposium. My name is Laurence Rijken, I'm the IDF Director General, and it's a pleasure to welcome you all to this event. This year's symposium focuses on Lactose 360 and is really looking at from the biology to the emerging insights. Before we begin, allow me to briefly introduce the International Dairy Federation. Founded in 1903, IDF is a global organization and it represents 41 member countries, accounting for approximately 2/3 of global milk production. One of IDF's unique strengths is its multi-stakeholder approach. And so we bring together experts from across the entire dairy value chain, including farmers, processors, academics, and governments. And this diversity allows IDF to provide science-based, consensus-driven guidance. Our work is supported by around 1,002 experts across 9 working areas, and nutrition and health is one of them. And so our mission is to nourish the world by advancing dairy knowledge. So let's turn to today's focus. So as part of their nutrient-dense package, dairy products naturally contain lactose. And so according to the World Health Organization, lactose is classified as milk sugar, which is a sugar naturally present in milk when it occurs within the structure of milk. And so in this context, lactose is considered intrinsic to the food matrix and is not classified as a free sugar. The definition of free sugars and related information can be found in this WHO guideline from 2015. So today's symposium really provides a comprehensive evidence-based perspective on lactose, from its biological role and digestion to its implication in health. Importantly, it will also address the distinction between lactose digestion, maldigestion, and lactose intolerance, which is commonly misunderstood. And throughout the years, IDF has been actively contributing to this area with its publications. And these resources aim to summarize current evidence on lactose as part of the dairy matrix and its role within healthy dietary patterns. But in addition to the publications we already have, I'm really pleased to announce that we're going to release as well a new publication, which is Understanding Lactose from Digestion to Health Outcomes. And this fact sheet explains what lactose is, its nutritional role in dairy products, and the evidence on its health-related properties, including prebiotic effects, glycemic response, and dental health implications. It also clarifies the distinction between lactose digestion, maldigestion, and lactose intolerance, emphasizing that unnecessary dairy avoidance can risk nutrient deficiency and really help position that conversation going forward. This symposium builds on the strong foundations of previous editions, and which have addressed topics such as dietary guidelines, the dairy matrix, and dairy across the life course. And together, these events contribute to an ongoing dialogue on the role of dairy in nutrition and health. And the recordings of these can be found on our IDF website. So within this event, we also have other events. And we have one annual flagship event, which is the IDF World Dairy Summit. This year, it will be hosted in New Zealand from the 15th to the 20th of November. And it really is a gathering of global leaders, dairy experts, scientists, and technical specialists to bring you the latest and the most significant issues facing the global dairy sector. As mentioned, IDF published diverse documents on a wide variety of topics in relation to the entire dairy chain, in addition to the nutrition one that I showed before. So I really encourage you to visit our publication webpage, where you will find other publications on nutrition and health, as well as other topics. You'll find the link in the chat, and you can see just a few examples in the slide. If you'd like to stay up to date with our work at IDF, sign up for our email newsletter on the IDF site. You can also find us on LinkedIn, X, and YouTube, where we regularly share updates on our work. I would like to take this opportunity to thank all speakers, moderators, and contributors, our experts, as well as staff who made this event possible. And I will now give the floor to Dr. Luz Maria de Regil, Director of the World Health Organization Department of Nutrition and Food Safety, who will provide the opening remarks. I'm wishing you a very good meeting today.
Good afternoon, good morning. Thank you so much for the invitation. I am delighted to address a community with deep scientific and technical expertise and a significant role in shaping what people eat and how foods are produced. We meet at a time of major changes in the global food systems. Countries in every region are confronting malnutrition in all its forms. We see rising diet-related non-communicable diseases, climate change, and persistent inequities. At the same time, there is increasing scrutiny on how foods are formulated, labeled, and marketed, particularly to children. Expectations from authorities and consumers are rising. They now extend not only to food safety and traceability, but also transparency, accountability, and sustainability. In this context, dairy remains highly visible as an important component of diets in many settings, and of course, as a source of livelihoods. We recognize the efforts of the dairy sector to ensure that food safety is non-negotiable for public health and consumer confidence. From primary production to processing, transport, and retail, robust food safety systems are essential to prevent illness, protect vulnerable populations, and sustain trust. At the same time, recent outbreaks associated with infant formula remind us that no system is immune to failure and that continued vigilance is required across all stages of production, including ingredients and manufacturing processes. On the nutritional side, dairy products can contribute important nutrients across the life course. However, from a public health perspective, we also need to address whether food products contain ingredients or nutrients of concern, such as free sugars, unhealthy fats, and sodium, that are associated with adverse health outcomes if consumed above the recommendations. This is particularly important as countries are calling for effective responses to raising obesity and cardiometabolic disease. In this equation or in this conversation, sustainability is an integral part of the discussion. Today, food systems are expected to deliver nutritious and safe foods while reducing environmental impact and strengthening long-term resilience. WHO is closely following innovation in this space and is also working on guidelines for animal source food consumption that consider the nutritional and environmental trade-offs to inform policy choices. The dairy sector is diverse, spanning countries, production systems, sizes, and markets. Our hope is that as a whole, it will rise positively to these challenges by strengthening food safety, improving food product profiles, supporting transparent labeling, advancing sustainability, and aligning marketing practices with public health goals, including adherence to the International Code of Marketing of Breast Milk Substitutes. The responsibility is significant, but so is the opportunity to strengthen trust, demonstrate leadership, and contribute constructively to healthier and more sustainable diets. I thank you for your attention and I wish you a very successful event. I look forward to our continued conversation.
Welcome, everyone, and thank you for joining us for the 6th IDF Nutrition and Health Symposium 2026. We are delighted to have you with us today. This year's theme is Lactose 360, Degrees from Biology to Emerging Benefits. Lactose continues to generate confusion among both consumers and health professionals. Although it is a naturally occurring sugar and a key component of dairy foods, worldwide, persistent misconceptions about lactose digestion, lactose intolerance, and its distinction from cow's milk allergy often leads to unnecessary dietary restrictions, despite the important nutrients dairy provides. The 6th IDF Nutrition and Health Symposium seeks to clarify these issues by presenting a full 360 degrees science-driven exploration of lactose from a health perspective, spanning physiology, clinical practice, emerging research, and practical dietary guidance. By sharing evidence-based insights from international experts, the symposium supports informed nutritional and health decisions and highlights the vital role of dairy in balanced, culturally diverse diets. I will be your moderator for this session too. My name is Seema Puri, and I've been a professor at the Department of Nutrition, Institute of Home Economics at the University of Delhi in India. Before we begin, some housekeeping announcements. This event is being recorded and the recording will be made available after the symposium. Secondly, please use the Q&A button to submit questions or comments as you watch the talks. We will bring them into the live panel discussion subsequently. Third, if you like subtitles, please turn on captions on your player settings to follow along during the presentations. I would also like to inform you that you can get your certificate of attendance or CPD certification upon request to the email given on the screen. The South African practitioners can earn a total of 2 CPD points for this event. German nutritional professionals receive 1 education credit for the eZert program. You need to scan the QR code for that. This IDF symposium can count towards continuing education for nutritional professionals in Canada also. You just heard from the IDF Director General and the WHO Director of the Department of Nutrition and Food Safety. This program also combines 4 pre-recorded talks with a live panel discussion, so you can benefit from both focused presentations and interactive expert exchange. Before we move on, let's make this session a bit more interactive with a quick audience poll. The same questions will be asked again at the end of the session to help us assess whether the presentation has influenced your perceptions. You will see 2 statements on your screen, and you will have to answer given the different options within a short period of time. So the first statement is lactose can play a functional role in health beyond simply providing energy. Please cast your vote now. Thank you. We will now come to the second question, which is generalized recommendations to avoid lactose are scientifically justified for most adults. Please cast your vote now. Thank you. We will now close this for voting. So for today's session, we have a fantastic lineup of speakers as shown on the slide. Our first speaker is Professor Corinna Walsh. A brief introduction about Professor Walsh. Corinna Walsh is a professor in the Department of Nutrition and Dietetics at the University of the Free State. Her research focuses on the development origin of health and disease, HIV, and malnutrition, with a particular interest in nutritional health, food security, and the impact of transitions from traditional to Western lifestyles on health. She serves on the Consumer Education Project of Milk South Africa Technical Advisory Committee and has a special interest in lactose. Corinna has published extensively and has supervised over 50 master's and doctoral students. She previously served on the board of the South African Medical Research Council and is the past president of the Nutrition Society of South Africa. So we can now have her presentation.
Good day everyone, and thank you to the International Dairy Federation for the invitation to participate in this symposium. Now, lactose is often discussed in the context of intolerance, and today our other speakers will explore this aspect of lactose and how to manage it in more detail. However, lactose is much more than a digestive challenge for some individuals. It's a biologically functional carbohydrate with important nutritional, metabolic, and physiological roles across the lifespan. So today I'd like to explore lactose from a broad nutritional and physiological perspective. In this presentation, I'll discuss lactose as a source of glucose and galactose for metabolic and neurological function, a facilitator of mineral absorption, a component of the dairy matrix that may contribute to broader health benefits, a contributor to musculoskeletal health, a potential regulator of satiety and appetite, and a low cariogenic carbohydrate. I'll also emphasize that many of these effects cannot be understood by examining lactose in isolation from the foods in which it naturally occurs. Then I'd also like to declare that I'm a member of the Technical Advisory Committee of Milk South Africa, and I have used Napkin as an AI tool to generate some of the images in my slides. Lactose is the principal carbohydrate in mammalian milk, and it consists of one molecule of glucose linked to one molecule of galactose. Human milk contains approximately 68 to 76 grams per litre of lactose, while cow's milk contains approximately 47 grams per litre. Traditionally, lactose has been viewed primarily as an energy source. However, emerging literature increasingly supports the idea that lactose has multiple biological functions beyond energy provision, and thus lactose should not simply be conceptualized as a dietary sugar, but rather as a functional carbohydrate within an evolutionary conserved food system. As I've mentioned, lactose digestion yields glucose and galactose. While glucose is widely discussed, galactose deserves greater attention. Importantly, lactose-derived galactose contributes to glycolipid synthesis, glycoprotein formation, cerebrosides, gangliosides, neuronal membrane structure, and potentially immune modulation. This becomes especially relevant during infancy, when rapid brain growth and myelination occur. These molecules are fundamental for nervous system development, cell signaling, and membrane integrity. During infancy, this may be particularly important because galactose contributes to the synthesis of cerebrosides and gangliosides that are involved in myelination and neural tissue development. Emerging evidence also suggests that lactose-derived galactose may support hepatic glycogen synthesis and exercise recovery. One of the most important concepts is that lactose has different physiological relevance across the life course. In infancy, we've already established that lactose supports energy transfer and contributes galactose for nervous system development. In addition, it also supports establishment of beneficial gut microbiota. During childhood and adolescence, lactose-containing dairy foods contribute substantially to calcium intake, skeletal growth, and nutrient adequacy. In adulthood, lactose contributes to nutrient density. It may influence satiety and supports healthy dietary patterns. In older adulthood, maintaining intake of dairy foods may help support bone and muscle health and reduce nutritional vulnerability. This becomes increasingly important with aging, where inadequate calcium and protein intake may contribute not only to osteoporosis, but also to sarcopenia and frailty. Dairy foods provide a coordinated package of calcium, phosphorus, and high-quality proteins that may help support musculoskeletal health across aging populations. Importantly, dairy foods provide substantially more calcium, protein, magnesium, potassium, and phosphorus per calorie than most other foods commonly consumed in adult diets. An important practical point over the whole lifespan is that unnecessary dairy avoidance may reduce the intake of calcium, vitamin B12, and high-quality protein, and this is especially important in populations that are already vulnerable to poor dietary quality. One of the longest recognized functional roles of lactose is its ability to facilitate mineral absorption. Evidence suggests that lactose enhances the absorption of calcium, magnesium, and potentially copper. Mechanistically, lactose hydrolysis and fermentation lower intestinal pH, improving mineral solubility and passive absorption. In addition, casein phosphopeptides generated during the digestion of dairy proteins may bind calcium and help maintain calcium in a soluble form within the intestinal lumen, thereby facilitating passive absorption in the ileum. Animal studies consistently demonstrate enhanced calcium absorption in the presence of lactose. The human evidence is strongest in infants, growing children, and potentially in individuals with lower calcium intake. In adults, the findings are more mixed, and this is most likely reflecting differences in lactase activity and dose, age and genetics, differences in habitual dairy intake, and the broader dietary context. Nonetheless, lactose-containing dairy foods remain among the most efficient dietary vehicles for calcium delivery. Now, previous IDF symposiums have highlighted the concept of the dairy matrix, and I'd like to highlight this crucial concept again today. So, foods are not merely collections of isolated nutrients. The food matrix is especially important in dairy research, where the health effects of milk, yogurt, and cheese cannot be predicted solely from their saturated fat, calcium, or lactose content in isolation. For example, fermented dairy products such as yogurt and cheese are consistently associated with favorable health outcomes, despite differing nutrient profiles. This suggests that interactions among nutrients, fermentation products, and the food structure play important biological roles. The health effects of dairy foods arise from interactions among lactose, proteins, fats, minerals, bioactive peptides, fermentation products, and food structure. The matrix influences digestion, nutrient bioavailability, satiety, glycemic response, and metabolic outcomes. Therefore, evaluating lactose outside the context of dairy foods risks oversimplifying its physiological role. This reductionist approach may partly explain why public perceptions around dairy and lactose often diverge from the broader evidence base regarding nutrient density and health outcomes. The evidence increasingly suggests that the health effects of dairy foods cannot be predicted simply from the effects of individual nutrients in isolation. Now, if we move on to bone health, we know that this should not be viewed as a calcium story alone. Increasingly, evidence supports a whole food and a whole diet approach to skeletal health. As we've mentioned, dairy foods provide not only lactose and calcium, but also other nutrients and bioactive peptides that interact synergistically within the food matrix. Lactose may contribute by improving mineral bioavailability within this matrix. The importance of this integrated nutrient package becomes particularly evident during periods of rapid skeletal growth. Randomized controlled trials in school-aged children have demonstrated higher gains in bone mineral content and bone mineral density among children receiving milk or dairy interventions compared with controls. Long-term avoidance of milk during childhood has been associated with lower bone mineral mass, smaller stature, and increased fracture risk before puberty. Furthermore, evidence suggests that dairy foods may be more effective for bone accretion than isolated calcium supplementation alone, potentially because of interactions among lactose, casein phosphopeptides, phosphorus, and protein within the dairy matrix. Thus, from a public health perspective, unnecessary dairy restriction can compromise nutritional health. Healthcare professionals, dietitians, and public messaging campaigns need to communicate carefully about lactose and dairy so people do not unnecessarily eliminate nutrient-dense foods, misunderstand lactose intolerance, or confuse lactose intolerance with milk allergy. An interesting emerging area concerns lactose and satiety regulation. Lactose has a relatively low glycemic index compared with many other sugars. Lactose has a glycemic index of approximately 46, which is substantially lower than that of glucose or sucrose. This slower glycemic response may contribute to more sustained energy availability, reduced glycemic excursions, and potentially improved satiety. Studies suggest that lactose-containing beverages may suppress hunger more effectively than glucose-containing beverages. Bowen and colleagues have demonstrated lower subsequent energy intake, lower appetite ratings, and prolonged ghrelin suppression after lactose consumption compared with glucose, and these may reflect slower digestion, slower absorption of galactose, and interactions between lactose and the dairy matrix. Additional work has shown that galactose ingestion results in lower postprandial glycemia and insulinemia than glucose ingestion and may reduce energy intake over a subsequent 24-hour period. Interestingly, broader evidence on dairy consumption suggests that dairy foods may support more favorable body composition profiles, including lower fat mass and preservation of lean body mass, particularly when these are incorporated into energy-restricted diets. Proposed mechanisms include enhanced satiety, altered fat oxidation, calcium-mediated reductions in fat absorption, and the effects of dairy proteins on appetite-regulating hormones. Importantly, these findings contrast sharply with a common perception that all sugars exert equivalent metabolic effects. Lactose is also unique from a dental perspective. Among common dietary sugars, lactose is consistently regarded as the least karyogenic. So compared with sucrose, lactose produces less acid, it causes a smaller reduction in oral pH, and creates a less favorable environment for karyogenic bacteria. Studies demonstrate that sucrose reduces oral pH below the critical threshold associated with enamel demineralization, whereas lactose generally does not. And animal and intraoral studies have consistently demonstrated lower karyogenicity of lactose relative to sucrose. In addition, dairy foods contain protective factors such as calcium, phosphorus, casein, lactoferrin, and lactoperoxidase. Thus, dairy foods may exert both lower karyogenicity and active protective effects within the oral environment. Importantly, the lower karyogenicity of lactose compared with sucrose reinforces the need to distinguish naturally occurring lactose within dairy foods from added free sugars in sugar-sweetened beverages and ultra-processed foods. Although not yet commonly emphasized in sport nutrition guidelines, lactose may also contribute to exercise metabolism. Recent work suggests that lactose can support carbohydrate oxidation during exercise, spare endogenous glycogen, and support post-exercise glycogen restoration. As I mentioned earlier, galactose appears particularly important for liver glycogen resynthesis. It's also been suggested that lactose has oxidation characteristics that are comparable to glucose during exercise. This is an emerging area, but it reinforces the idea that lactose should not be dismissed simply as a sugar with digestive implications. Instead, it may represent a useful functional carbohydrate under specific physiological conditions. And this may be particularly relevant for recovery nutrition, where dairy foods provide not only carbohydrates in the form of lactose, but also high-quality proteins and electrolytes that support post-exercise recovery and muscle protein synthesis. So in conclusion, lactose is far more than milk sugar. It's a functional carbohydrate, it's a source of glucose and galactose, a facilitator of mineral absorption, a low karyogenic sugar, and a potential contributor to satiety regulation. Also part of the broader dairy matrix that supports nutrient-dense diets. Across the lifespan, lactose-containing dairy foods contribute to nutrition and health in ways that extend well beyond energy provision, supporting skeletal development, nutrient adequacy, metabolic regulation, and the consumption of balanced nutrient-dense diets. Ultimately, lactose is part of a nutritional system in which carbohydrate, protein, minerals, and bioactive compounds interact within the dairy matrix to support human health. As nutrition scientists and health professionals, we should continue moving beyond reductionist views of single nutrients and towards understanding foods within their biological and dietary context. The following two slides indicate the references that I have used for this presentation. Thank you very much for listening, and I look forward to engaging with you during the panel discussions that will follow.
So we've had a very illustrative talk by Dr. Corina Walsh. She highlighted the multifaceted physiological role of lactose. Besides energy, it has very important roles to play in bone health, dental health, body composition, physical performance, appetite regulation, satiety, and many, many more. We'll now go on to the next talk, which is given by Professor Michael Ganzel. Dr. Michael Ganzel is trained as a food engineer at the University of Hohenheim in Stuttgart, Germany, and has obtained his doctoral and postdoctoral degrees in food microbiology at the University of Hohenheim and the Technical University of Munich, respectively. He is professor and Canada Research Chair in food microbiology and probiotics at the University of Alberta, Edmonton, Canada, and was elected Fellow of the Royal Society of Canada in 2023. From 2021 to 2024, he was recognized as the Clarivate Highly Cited Researcher. His research links microbial phylogeny, physiology, and ecology to food quality, food safety, and host health. So we'll now have Dr. Ganzel's presentation.
Welcome to the IDF Symposium on lactose. My name is Michael Ganzel. I'm teaching food microbiology at the University of Alberta in Canada, and will share my thoughts on health beneficial and adverse health effects of lactose in the next 20 minutes or so. Let me start by sharing my screen. This is a little bit bigger. So the question is, health effect of lactose, is it anti-nutritive, health beneficial, or is it both? The nutritional functionality of oligosaccharides is a little bit complicated. I will take you through a walk in the supermarket. If you go to the dairy section, you will find fluid milk, which obviously contains lactose, but you will also find lactose-free milk because many individuals perceive lactose as something which provides adverse health effects. If you go a little bit further, you find beta-galacto-oligosaccharides, which are structurally very similar to lactose as a health beneficial ingredients in food, particularly infant formula. So they are bifidogenic and probably are good. If you go to the pulse section of the supermarket, you find soy milk or beans or chickpeas with raffinose family oligosaccharides, which are known to provide adverse health effects in all individuals, but also have been rebranded as vegan GOS with beneficial health properties. For beta fructans, you have the same situation. In one section of the supermarket, for example, in the dairy products, they're used as health beneficial ingredients to stimulate intestinal bifidobacteria. In other sections of the supermarket, at least in Canada, you will find low FODMAP products where fructooligosaccharides and alpha-galacto-oligosaccharides were removed enzymatically to avoid adverse reactions. Before dwelling further into lactose, let's look at the lactose content of dairy products. It's highest in fluid milk with approximately 40 grams per liter. Fermentation doesn't remove all that much of the lactose. So in yogurt, strained yogurt, or sour milk products, you still have 30 to 36 grams per lactose. In butter and in cheese, most of the lactose in milk is removed with the buttermilk and the whey. So in butter and in ripened cheeses, the lactose content is very low. In fresh cheeses, such as cream cheese, it's substantially higher because whey removal is not quite as substantial. Products printed in bold are well tolerated by individuals with lactose intolerance because the products come with microbial beta-galactosidases, which hydrolyzes lactase. I have a question mark behind all products fermented with mesophilic starter cultures. First, because the biochemistry in these products is different. Most mesophilic cultures hydrolyze lactose with phospho-beta-galactosidases, which are not active on lactase after cell death. And second, the clinical studies with sour milk or sour buttermilk are essentially lacking. We clearly have to take a closer look at oligodigestion in the small intestine. Not much happens with lactose in the oral cavity because these organisms prefer sucrose and simple sugars. In the small intestine, oligosaccharides are hydrolyzed by brush border oligosaccharide hydrolases. And humans have an enzyme which is called maltase glucoamylase, active on alpha-glucans, sucrase, isomaltase, which is active on sucrose and alpha-1,4-linked alpha-glucooligosaccharides, and lactase fluorescein hydrolase, which hydrolyzes lactose, but also beta-glucosylated phytochemicals, including soy isoflavones and trehalase. Whatever is not hydrolyzed by brush border enzymes, either because the oligosaccharide supply is greater than the enzyme activity or because the enzyme has gone missing, is fermented by bacteria in the small intestine. These differ from colonic microbiota because facultative anaerobes dominate, including lactic acid bacteria and Enterobacteriaceae and bifidobacteria. What happens to undigested lactose? Let's start with infants. All human infants express brush border lactase. Nevertheless, a substantial proportion of the lactose reaches the colon to be fermented by colonic microorganisms. How much is transferred depends on the individuals, so each infant is different. It also depends on the age. Preterm infants with a very immature intestinal digestive system, much more than 50% of the lactose can be fermented. As infants age, most of the lactose is hydrolyzed in the small intestine, and only a small proportion of the lactose is fermented in the colon. Nevertheless, lactose is an important driver for the development of infant microbiota. This is a study which compares two breastfed infants with infants with a cow milk allergy. For the breastfed infants, lactose is the major carbohydrate source in the diet. And as is well described, bifidobacteria are among those that benefit most from the availability of lactose as a sugar supply. In infants with a cow milk protein allergy, there are no cow milk proteins in the formulae. There is also no lactose, and correspondingly, the proportion of bifidobacteria, particularly those species which are typical in the intestine of infants, is very low, and other bacteria dominate, which means in these infants, pathogen defense is much less likely. It is well documented that human milk oligosaccharides stimulate intestinal bifidobacteria in infants. What is the relevance of lactose relative to human milk oligosaccharides? The answer is these two act synergistically. This is an in vitro study which looked at in vitro fecal fermentations with a stool of human donors. And human milk oligosaccharides stimulate intestinal formation of acetate, lactate, and total short-chain fatty acids after 6, 24, and 48 hours. Lactose is actually a lot more effective than human milk oligosaccharides. And you get the best result if human milk oligosaccharides is combined with lactose, because intestinal bifidobacteria, which are important in infants to provide defense against enteric pathogens, are very good at metabolizing lactose, but they're also perfectly capable of metabolizing human milk oligosaccharides. Now, is lactose a digestible or an indigestible oligosaccharide? It depends. In 70% of human adults, that's the ancestral wild-type state, lactose is indigestible because the expression of brush border lactase fluorescein hydrolase is decreased during growth into adulthood, and these individuals are lactase non-persistent. That term designates the genotype. The phenotype is designated by the term lactose intolerance. Keep in mind that the genotype and the phenotype do not completely overlap. Lactose is not the only conditionally digested sugar. In about 15% of humans, fructose is indigestible because it's poorly absorbed. These individuals are at a much higher risk of irritable bowel syndrome. On a global perspective, a very small proportion is sucrose intolerant. I emphasize that in my lecture, nevertheless, because 5 to 10% of the Canadian Inuit population is sucrose intolerant. These individuals are also at a higher risk of irritable bowel syndrome. And many of my students become dietitians. And if they practice in the north, they will have to recognize sucrose intolerance. The effect of indigestible oligosaccharides is entirely dose dependent. This is a study from Japan, where a vast majority of individuals is lactase non-persistent. What you see here is the dose of the sugar intake plotted against the cumulative incidence of diarrhea. The paper is a bit old, but it has aged very well. Up to approximately 0.3 gram of sugar per kilogram of body weight, everything is happy and well. And then you see an increasing incidence of adverse intestinal events, including diarrhea. If you compare different non-digestible oligosaccharide, let's ignore the partially digestible oligosaccharides in this purpose, lactose, salobiose, galactosyl lactose, and fructooligosaccharides. The story is pretty much the same for all of those. You end up at a dose of about 0.3 gram per kilogram body weight, which translates approximately 15 gram per day of non-digestible oligosaccharide. And this number is very consistent with the maximum level of oligosaccharides that is used in prebiotic studies with GOS and FOS. Nobody ever uses more than 15 gram per day because the people know that the patients would explode. Among the major oligosaccharides that are used for food applications, term prebiotic, I don't like the term, I can explain that in the panel discussion that is to follow, are galacto-oligosaccharides, fructooligosaccharides, inulin, and resistant starch. With these four, you probably capture 80% of the market. And FDA considers all four as dietary fiber because they meet the WHO definition and physiological benefits were documented in clinical studies. What happens if you overdose? You get accumulation of water and gas in the small intestine, which results in osmotic diarrhea and bloating. This is a study which used fructose in fructose malabsorbing individuals. You see here the curve depicting water accumulation relative to glucose, breath hydrogen relative to glucose, which indicates that you have fermentation. If the study would be done with lactose, the result would likely look very similar. Oh, what happens now? Sorry, I will now stop share. I made a wrong mistake on my PowerPoint slide. I was here. Okay, now I can continue. Apologies. Where do those individuals with predominant lactase persistent genotype live? Mostly in regions of the world which are either too cold and too wet, like in the north and northwest of Europe, or in regions of the world which are so dry that you cannot grow crops other than grass, and you need ruminants to convert the grass into something which is useful as dietary energy for humans, and drinking milk is the most efficient way of gaining dietary energy from ruminants. So the ability to digest lactose greatly increase the reproductive success and increase the proportion of lactase persistent individuals in these populations to more than 90% in a few hundred generations. In lactase persistent individuals, lactose is hydrolyzed in the small intestine and taken up as a sugar. In lactase non-persistent individuals, lactose is transported into the colon and fermented by colonic microbiota. And I mentioned earlier that bifidobacteria is an undisputed champion of efficient lactose utilization, which means bifidobacteria are those that profit the most from the supply of lactose to the colon. One of the first studies that made the case, many made the case afterwards, was a genome-wide association study with a large cohort in the Netherlands, which aimed to link the genotype of humans to the composition of the gut microbiota. The study reported very few convincing correlations, among the few was the gene or the mutation causing lactase persistent in lactase non-persistent individuals, which in the Netherlands presumably still consume a lot of dairy products. The intestinal abundance of bifidobacteria was substantially higher than in lactase persistent studies. And this was confirmed by a second data set. So bifidobacteria increase if lactose is consumed by lactase non-persistent individuals. This was confirmed in multiple other studies that were published afterwards. This is one example where a probiotic strain of Bifidobacterium longum was administered for 2 weeks, and then the individual study participants were followed for 9 months whether the strain persists. And about approximately half of the study participants, the ecological niche in the colon, which is available for lactose-consuming microorganisms, was occupied. So here the incoming human-adapted probiotic strain did not persist. And the other half of individuals where the niche was not occupied, very few bifidobacteria initially present, the niche for a lactose-consuming bifidobacterium was open, and this strain of Bifidobacterium longum persisted for more than 9 months after probiotic intervention was stopped. Let me start to summarize the presentations by looking at the health benefits. If we look at health benefits of non-digestible carbohydrates, including lactose in lactase non-persistent individuals, the karyogenicity is low. They have a reduced caloric content because they're fermented, and fermentation provides dietary energy by short-chain fatty acids, but that's only 60 to 75% of the glucose. You have a low glycemic index, which is for lactose irrespective of the genotype, because even in lactase persistent individuals, half of the molecule is glucose, which doesn't contribute to an increase of the glycemic index. And fermentation generates short-chain fatty acids by fermentation of colonic microbiota, which is considered to be entirely health beneficial. What happens if the dose exceeds more than 0.3 gram per kilogram body weight, or approximately 15 grams per day? Fermentation draws water to the small intestine and leads in excessive gas formation, and now diarrhea and intestinal bloating ensue, which obviously we don't think is very pleasant. Main bottom line, oligosaccharide intolerance and prebiotic activity are two sides of the same coin, and the two are separated only by low dose versus high dose. This is one of 3 good reasons why I all but eliminated the use of the term prebiotic in any of my teaching and writing. And we need to take care. The genotype is fixed, but adverse effects of non-digestible oligosaccharides are very different among individuals, and they can change over time because the ability of intestinal microbiota to ferment non-digestible oligosaccharides without adverse effects can change depending on disease, antibiotic use, and other life events. Let me take that back to the dairy products. How much of the dairy products can we consume? If we assume that a threshold of 15 grams per lactose a day are tolerated by anyone, for fluid milk, we have a limit of 375 milliliters per day. For sweet buttermilk, the limit is approximately the same. For all other dairy products, particularly the fermented dairy products, the level is a lot higher, to the point that anyone can consume as much as they want, either because lactose is removed by whey or buttermilk, or because fermentation means that the lactose comes with a beta-galactosidase, which is active during small intestinal transit and eliminates all negative health consequences, with a small question mark, because for products which are produced with mesophilic starter cultures which utilize lactose by phosphobeta-galactosidase, the mechanism can be different, and the number of clinical studies is too low to afford conclusions. The summary: with exception of fluid milk, most dairy products are tolerated independent of lactase persistence. If you happen to be lactase non-persister, you have access to a very cheap source of prebiotics which increase intestinal abundance of bifidobacteria. Fluid milk or yogurt will do the job just fine. And if you look at the bigger scheme of things, by looking at which part of the diet is health beneficial, and which part of the which dietary items are not beneficial, low-fat dairy products, and in this category, particularly low-fat fermented dairy products, such as yogurt, have a very good reputation of being health beneficial, both because of the matrix properties, milk, which comes with lactose, which comes with calcium, and that is very much bioavailability, and because of the live dietary microbes, which appear to have some probiotic activities. So replacing dairy products, particularly fermented dairy products, with plant-based products may not always be a health beneficial choice. With that, I want to thank my graduate students, which have done great things in terms of understanding lactose metabolisms by food fermenting bacteria and in the intestine. I want to thank my students in the undergrad classes, because teaching the students has taught me how to relate the topic. And with that, I will end the presentation to be available for the live panel discussion that ensues. Thanks for listening.
So that was a very detailed presentation, and the message that has come across very clearly is that, with the exception of milk, most dairy is tolerated independent of lactase persistence. And there's a strong recommendation for low-fat dairy products, especially fermented products, which have innumerable health benefits. So going on to the next presentation, which is by Assistant Professor Andrew Silagi. Dr. Silagi graduated from medicine at the McGill University in 1974, completed internal medicine in Montreal. He is a trained gastroenterologist who completed 3 years at the Tufts Gastroenterology Program in Boston, USA. He returned to Montreal and ultimately worked as a full-time gastroenterologist at the Sir Mortimer Jewish General Hospital as an assistant professor of medicine. His duties included clinical management of patients, investigation of their problems with endoscopy, teaching of residents and students. He also conducted clinical research projects. His interests gravitated towards problems related to lactose because of the development of the process of bacterial adaptation. He is currently in the process of retiring from clinical work, but has retained interest in keeping up with the progress on lactose. So we'll now hear the presentation by Professor Andrew.
Should the role of lactose be reconsidered in adults with loss of ability to digest? Lactose is a disaccharide which is an important neonatal nutrient. This consists of 2 aldohexoses, a D-galactopyranosyl joined in a 1-to-4 beta-glycoside linkage to D-glucose. The difference between these 2 molecules is in position carbon 4 of galactose, where the hydroxyl radical points upwards. The digestion of lactose is virtually 100% in neonates through the enzyme lactase, which is also called lactase fluorescent hydrolase, a beta-glycosidase or beta-galactosidase, and is a product of the LCT gene. The disaccharide is digested into its monosaccharide form, glucose and galactose, and is transported across the brush border by the sodium-coupled glucose transporter, at which point glucose is utilized entirely for energy or stored for later use as glycogen in the liver. Galactose, on the other hand, has to be detoxified by the 4 enzymes of the Loire pathway, and then can be utilized also for energy or later use as glycogen, but it also functions for multiple developmental functions like the brain, immunity, and others. There are minor metabolisms of lactose as well through reduction and oxidation, as well as a small amount of endogenous production, which is inversely related to age. The LCT gene is located on the long arm of chromosome 2 and consists of 17 exons with a span of almost 50,000 kilobase pairs. The human lactase DNA encodes for a polypeptide of 1,927 amino acids, which is exclusively located on the small intestinal brush border and primarily in the villus tips, with the highest concentration in the jejunum as opposed to the duodenum. The gene, however, is activated by a transcription promoter which is located at the 5' end near the LCT gene, and also an enhancer region which is situated about 14 kilobase pairs upstream in the cis position, same side. The enhancers are situated in intron 13 mainly of the gene min chromosome maintenance components 6. Other introns also are sources of transcription controllers. Following weaning, in the majority of the world's population, enzyme levels sink to 5 to 10% compared to that at birth. The interesting part is that the control of degradation of the lactase enzyme depends on mainly epigenetic control. It consists of both hyper-DNA methylation of the recessive allele and hypomethylation of the dominant allele. This is carried out by transcription factors, which define the various single nucleotide peptides of the lactase gene. These factors, these transcription factors, are also governed by genes. One of the most important ones is so-called octamer binding protein 1 and binds very tightly to the dominant T allele of the 13-9-10 single SNP and much more loosely to the CC recessive allele. This allows for translation, continuous translation of the TT allele, but because of DNA methylation, it reduces translation of the CC allele, leading to the reduction in lactase enzyme levels. These degradations begin at various ages throughout the world. Polymorphisms of lactase are listed on this slide. These 5 represent the most common genes which have been studied. The first one, which was described by Anata's group from Finland, was the cyasin to thymidine transformation of position 13-9-10 at the MCM6 mini chromosome. This is the primary gene controlling lactase persistence in North Europe, North America, Central Asia, India, and Australia. The other 4 have been described in mostly Africa and Middle East by the people that are designated here. And basically these genes appeared more or less around the same time, from 7 to 10,000 years ago, by a process called convergent evolution or culture coevolution. There is an additional gene, which is listed at the bottom in the footnote, which accompanies the European version of lactase controller, but it doesn't work by itself. It has to be associated. So it may be an enhancer of the European version. This map outlines the world distributions of lactase persistent in red and shades of lower colors, blue, lactase non-persistent. This graph came from the end of the last century mostly, and it should be noted that at the end of the last century to the beginning of this century, lots of migrations occurred from LNP to LP populations, and the map certainly will change. The terms related to adult lactose consumption are divided into three, which probably shouldn't be mixed up. The classical genetic distinctions are lactase persistence, lactase non-persistence. The second two are related to enzyme, either deficiency and maldigestion. Again, these are independent of genetics. And then the last group are symptoms, which should certainly not be confused with those related to enzymes. Lactose intolerance basically applies to LNP populations. On the other hand, self-reported lactose intolerance are those persons who believe themselves to be lactose maldigesters but have not been tested. When they are tested and found to be LP, they're called functional lactose intolerance, and when they are sensitive, non-specifically called lactose sensitivity, which was first described in inflammatory bowel diseases but applies to non-patients with IBD as well. Maldigestion of lactose is defined by four groups. There's the congenital lactase deficiency, which is a rare variant or mutation in the lactase gene itself, and basically has been described rarely in the Japanese and Finnish population. In the Finnish population, it represents about 1 in 60,000 births. Developmental lactase deficiency, on the other hand, is more common and occurs in premature neonates and some term neonates, and it has been established that lactase fully develops in neonates by 5 to 7 days postpartum. The main cause of lactase deficiency, however, in adults is the genetic differential, which divides the world into those who can and those who cannot digest lactose, based on approximately 1/4 to 1/3 of the world's population who are dominant able to digest lactose, the rest cannot. Secondary lactase deficiency is independent of genetics and occurs when either toxins, anatomical redistribution, or certain diseases, as listed on the side, interfere with absorption of and digestion. Symptoms related to lactose. The symptoms of lactose intolerance consist of both intestinal and extra-intestinal manifestations. The intestinal manifestations of gas, bloat, abdominal pain, diarrhea, sometimes vomiting, and the extra-intestinal manifestation of headache, fatigue, and mood disorders are not specific to lactose intolerance, but may be found in any sugar intolerances and number of syndromes as well as diseases. On the bottom of the slide are listed some of the more common causes of symptoms: celiac disease, irritable bowel syndrome, and a number of other sugar intolerances, which are listed here and can be read in the literature. Other causes of symptoms which are associated with milk and dairy consumption can be attributed to other things besides lactose. The most interesting one is the recognition that cows from North Europe or South Europe have a genetic makeup which is different in the production of beta-casein. And this is labeled A1 from Northern European cows and A2 from other genetic origin of cows. The beta-casein from A1 cows or A1 milk substitutes histidine for proline at position 67 and is associated with the production of a specific beta-casomorphin 7 protein, which is an opioid agonist, which interacts with small bowel opioid receptors, resulting in gastrointestinal symptoms. Other potential cause includes fats, like particularly in ice cream, but others as well. Alpha-glycerophosphate can lead to symptoms resembling lactose intolerance. Irritable bowel syndrome is one of the so-called functional gastrointestinal disorders with a high frequency of self-reported lactose intolerance, as well as increased sensitivity to lactose. The last two are diseases. They're allergies to protein in milk, like casein, whey, and lactoglobulins. In children, the disease or the allergy to milk resembles ulcerative colitis with bloody diarrhea. In adults, the GI or gastrointestinal manifestations are reduced. The most recent disease associated with an autoimmune reaction to a number of food allergy antigens, but particularly milk, is eosinophilic esophagitis, which is a specific disease of the esophagus resulting in a number of symptoms. And in fact, one of the treatments is to withdraw milk. The fate of lactose in lactase non-persistent population. Lactose intolerance in lactose maldigesters. Not all LNP persons are lactose intolerant, and in fact, the rate of lactose intolerance and self-reported lactose intolerance are similar in frequency between both LP and LNP populations. On a physiological level, excessive lactose ingestion in single doses that are large enough induces an osmotic load, which results in cramps, diarrhea, and gas. He et al. Showed that bacterial metabolism can be quite rapid and quantitatively increase the production of short-chain fatty acids, which are then affecting again the symptomatology in the colon. Metabolomic studies were also done, which distinguished between both LP and LNP lactose intolerant and LNP lactose tolerant. Windy et al. Found that the main chemical that was different between LP and LNP was 5-methylfuran carboxylaldehyde. And the difference between LNP lactose intolerant and LNP lactose tolerant was identified as acetaldehyde and other fatty acid aldehyde. Variables and conditions which could modify symptoms of lactose intolerance in lactose maldigesters. Consuming lactose containing products with other foods, especially fats, or for example yogurt with a high viscosity, can alter symptoms of lactose intolerance. If there is medications taken before lactose, and these medications alter in prolonged transit times of the gut, symptoms can be reduced. In pregnant women who are lactose maldigesters, progression of pregnancy improves their lactose intolerance, either via the effects of progesterone on intestinal prolongation of transit time and or adaptation to increasing quantities of dairy which can be consumed. Certain diseases which alter gastric emptying and intestinal transit also will have a modification effect on lactose intolerance. Certain diseases, particularly hyperthyroid, possibly hypothyroid, which both alter the transit times rapidly with hyper and slowed with hypo may have a similar effect in reducing or increasing symptoms. In addition, thyroid hormones have been suggested to inhibit lactase at the intestinal level. Certain other diseases which affect the villus enzyme levels and other diseases which have an effect on transit time also modify symptoms. Lactose metabolism in lactase non-persistent populations. It was reported that the minimal dose which reaches the colon in LNP populations approximately 6 grams. The maximum dose of lactose which reaches the colon in LP populations is about 8% of ingested dairy foods containing lactose. This is equal to approximately 4 grams per liter of milk. It was also reported in the end of last century that tolerance to 1 or 2 cups of milk divided in LNP populations is easily tolerated. And furthermore, continued regular consumption of lactose in LNP populations, but not LP populations, leads to beneficial microbiome changes. Fermentation of lactose by colonic bacteria which possess beta-galactosidase proceeds initially as it does in the small intestine. That means lactose is broken into glucose and galactose and then is further fermented through pyruvate and the gases of hydrogen, carbon dioxide, a number of short-chain fatty acids. Then secondary and tertiary bacteria use these products to form the end stage of fermentation into methane, acetate, butyrate, and propionate, of which acetate and propionate are used primarily for caloric salvage, and butyrate, in addition to supplying colonocytes, is important in both inflammatory control and anti-neoplastic effects. This slide outlines the number of reports in the last 3 decades which support the observation that lactase non-persistent populations can adapt by consuming regular amounts of lactose. I will not go over these in great detail except to point out that epidemiologically the Japanese have done most of the work on this and have shown in a study that bifidobacterial population expansion in these patients basically correlated positively with the amount of dairy food that was taken to consume. Most recently, the prebiotic potential of lactose was shown in lactase persistent populations by introducing small quantities of lactose into a colon model in vitro, which resulted in the expansion of lactobacilli and a number of other beneficial bacteria. These small doses were representative of the previously published 8% maximum spill of lactose into the colon of lactase persistent populations. What this study showed also is why lactose is a selective prebiotic in LNP, because the LP populations are unable to increase significantly the amount of lactose that they spill because they digest it in the small intestine. What are the ramifications of lactose adaptation, which is often overlooked when evaluating diseases for dairy foods? Many diseases have been studied for effects of milk and dairy foods. A recent scoping meta-analysis of 29 studies showed that little over 1/3 were associated with a moderate reduction in risks, whereas only 4.3% were associated with increased risk for such diseases as prostate, testicular cancer, and perhaps Parkinson's disease. However, colorectal cancer has been the most studied for the effects of dairy foods, and most of them agree that there's a modest decreased risk for colorectal cancer. However, multiple confounding factors plague these studies, including the sites of cancer reduction in the colon, the types of dairy foods that are involved, and which nutrients are the effects. However, there is one confounding factor which has not been seriously considered. This slide shows the relationship between a superficial layer of epithelial cells called colonocytes, a layer of mucus mixed with bacteria called the biofilm, and the luminal side, which are representing the microbiome. This slide also shows a number of nutrients in dairy foods which are involved with anti-neoplastic, anti-inflammatory effects, such as calcium, conjugated linoleic acid, folic acid, and lactoferrin. However, I will concentrate on the relationship with the microbiome. It's important to note that in parallel with the discovery of adaptation to lactose, much work has shown that the microbiome is intimately related to the pathogenesis of a number of different diseases. The beginning of these diseases often begins with a disorder in the microbiome from what we call eubiosis to dysbiosis. Dysbiosis is defined as a loss of diversity, richness, a loss of beneficial bacteria, and increase in potential pathogens called pathobionts. When this happens, there's an effect on the biofilm as well as on the glue or proteins which hold the epithelial cells together. These tend to loosen and allow for the passage of bacteria and bacterial products to traverse into the host and incite a chronic inflammatory reaction, which together with genetics and environmental exposomes potentially could lead to different kind of diseases. It's been shown that it would be desirable to reduce the dysbiotic microbiome and return it to a eubiotic state, because this would reduce the passage of bacteria into the host. And this slide outlines the number of different diseases which are putatively related to alterations of the microbiome. These include not only gastrointestinal diseases, but also cardiometabolic diseases, obesity, allergies, and neuropsychiatric diseases. So the practical consequences of colonic adaptation is increased capacity to consume dairy foods with improved symptoms. The probiotic type of bacteria may contribute to reduction of dysbiosis or prevention of dysbiosis. Unfortunately, the frequency at this time of occult lactose-adapted persons is unknown, and it may be very relevant in mixed populations where milk drinking is culturally acceptable. So the corollaries of this is that genetic predisposition or genetic prediction of milk and dairy food effect on diseases may be inappropriate in mixed populations. The use of Mendelian randomization, which is a statistical method to reduce confounding in observational studies, may be inaccurate because bacterial digestion as opposed to intestinal digestion introduces a horizontal pleiotropy, which reduces the accuracy of Mendelian randomization. Thus, the analysis of LP, LNP adapted populations together also raises the confounder of Simpson's paradox in statistic depending on the proportion of LP, LNP distributions. Simpson's paradox is a mathematical phenomenon where a trend appears in different groups of data but disappears or even reverses when these groups are combined. We can now reassess as to why lactose is important, and we can divide humanity into three functional groups. The first group are neonates, where close to 100% contain or have the enzyme lactase and are able to digest lactose. Group 2 are the older children and adults, where the great divide takes place. And group 3 are lactase non-persistent populations who adapt by regular lactose consumption and remain hidden within the larger group of dairy-consuming populations. So the take-home message from this is that the notion that lactose is to be avoided in few foods that it represents a pariah of nutrients needs to be decreased and emphasized. On individual level, dietitians should be familiar with the nuances of dairy food consumption and how it fits in the management of functional and other gastrointestinal disorders. On research level, the establishment of adaptation should inspire clinical trials as human models of the effects of natural probiotics on different diseases. And finally, the evaluation of potential effects of dairy foods on diseases should take into consideration the proportion of LNP adapted persons in such studies, because failure to do so may reduce true effects. Thank you.
Dr. Silagi's presentation, I think the last sentence did resonate very strongly with me, which said that the notion that lactose has to be avoided is misdirected. And we really need a deeper understanding into the nuances of dairy food consumption. And I think that's a very strong message which has come across from his presentation. Going on to the last but not the least presentation by Professor Miranda Loma. Miranda Loma is a senior consultant dietitian in gastroenterology at Guy's and St. Thomas's Hospital, London, and has an adjunct appointment as Professor of Dietetics in Gastroenterology at King's College, London. Professor Loma qualified as a dietitian in 1990, and in 2002, she completed a PhD on diet and Crohn's disease. Her research interests are dietary assessment and nutritional management of gastrointestinal disorders, including inflammatory bowel disease and disorders of the gut-brain interaction. She has expertise in writing guidelines, conducting multicentric clinical trials, and service delivery. Professor Loma is editor of a book entitled Advanced Nutrition and Dietetics in Gastroenterology. She gave the BDA, British Dietetic Association's Elsie Widdowson Lecture in 2014 as recognition of her international impact in dietetics. In 2016, she was awarded an MBE for services to dietetics and gastroenterology. So let's move on to her presentation.
Hello, my name's Miranda Loma. I'm a consultant dietitian in gastroenterology at Guy's and St Thomas's in London and a professor of dietetics in gastroenterology at King's College London. My interests are the dietary management of irritable bowel syndrome and inflammatory bowel disease, and today I'm going to be talking about the dietary management of lactose intolerance. These are my conflicts of interest. And today I'm going to cover diagnosis of lactose malabsorption and intolerance, dietary management strategies for lactose intolerance, the lactose content of common dairy foods, and the overlap of lactose intolerance with irritable bowel syndrome, and some practical strategies to ensure nutritional adequacy and overall diet quality. Thinking about the terms lactose malabsorption and lactose intolerance. These are both very different. Lactose malabsorption means the incomplete hydrolysis of lactose and the presence of unabsorbed lactose in the colonic lumen, whereas lactose intolerance is the generation of symptoms in relation to that unabsorbed lactose being present in the colonic lumen, and it's very dependent on self-reported symptoms after lactose ingestion. What's important to note is that not everybody with lactose malabsorption has lactose intolerance, and also vice versa. Symptoms of lactose intolerance would include things like abdominal pain, bloating, flatus, wind, diarrhea, borborygmi, nausea and vomiting, and constipation. If we look at the pathophysiology of lactose intolerance, you can see on the left-hand side normal lactose digestion, where lactose is broken down by the enzyme lactase, resulting in the production of glucose and galactose, which gets absorbed into the bloodstream. Whereas in lactose intolerance, there isn't any of that lactase, and what happens is you get an increase in the water occurring within the gut, being drawn in by the lactose itself, and this can increase motility through the gut, and the lactose also ends up being present in the colonic part of the gut, and this gets fermented by the bacteria and causes acids and gases, and that can cause the symptoms that are associated with lactose intolerance. What's really key is how do we diagnose lactose malabsorption and how do we diagnose lactose intolerance? So lactose malabsorption, there are lots of different tests that we might be using. So there are direct tests and indirect tests. The most common test that we might use for measuring lactose malabsorption is the breath hydrogen test. And with this test, what happens is that we measure a load of, measure hydrogen and methane after a load of lactose for 3 hours, and lactose digesters do not increase the breath hydrogen level, and it's the most common clinical test that we might be using. Just to show graphically what happens is once you take the lactose, usually a dose up to 25 grams on an overnight fast, and making sure that there are no other fermentable carbohydrates in the diet the day before. The lactose is consumed and then it passes through the small intestine. If it gets absorbed, then no lactose will impact the large intestine. But if the lactose isn't absorbed, then it gets fermented by the bacteria, producing hydrogen and also methane. And this hydrogen then gets absorbed into the bloodstream and it gets exhaled via the lungs. So we measure the breath hydrogen level. There are variations in breath testing protocols. There are pre-test instructions. People shouldn't have antibiotics or prebiotics for 4 weeks before the test, and no bowel preparation in the previous 4 weeks either. They shouldn't have had any laxatives or antidiarrheal medication for 3 days, and they should avoid fermentable carbohydrates for 24 hours, as I just mentioned earlier. The fast is usually overnight, and they should have no chewing gum or smoking pre the test. So there's a lot involved in making sure that these tests are done correctly. And what happens is at baseline, you take a breath sample, ingest the test liquid, as I said, 25 grams of lactose in usually about 250 mils of fluid, and then breath samples are taken at 15 to 30 minute intervals until 3 or 4 hours. Symptoms are recorded throughout the test to see if there are any signs of intolerance, and as I said, protocols do vary. The breath sampling intervals may vary and the diagnostic criteria may vary. What's also important to note is that often people might have a lactulose breath test in advance of the lactose breath test, and the reason for that is to see if hydrogen and methane are being produced after the consumption of lactulose, which is a non-absorbable carbohydrate. So coming back to these terms again, lactose malabsorption and lactose intolerance. I think what we need to be aware of is the test that we've just described is really looking at lactose malabsorption, whereas lactose intolerance is far more difficult to identify. And I did mention that it's important to describe any symptoms that occur during the lactose malabsorption test, because this would give us an indication of whether there is any signs of intolerance. A way to diagnose lactose intolerance is really to use the gold standard dietary exclusion method. And this would be where we would exclude dietary lactose until symptom resolution, and then re-challenge with lactose to induce symptoms, and then finally to reintroduce lactose to a personal tolerance threshold. Now this gold standard test will really take several weeks, because excluding lactose, it may take a few days or even up to a week for the lactose symptoms, the lactose intolerance symptoms to resolve. And then it may take a while to sort of think about when is the best time to re-challenge to induce symptoms. And once that's done, we then need to be thinking about how long it's going to take to reintroduce lactose to the personal tolerance threshold level. In terms of the management of lactose intolerance itself, we only suggest restriction of lactose if it's absolutely necessary. And I mentioned that it may take just a week or so for symptoms to resolve, but often it might be that we would exclude lactose for up to 4 weeks. But I would certainly suggest that you're not doing it for longer than that. You do want to monitor symptoms during that time, and when it comes to that whole reintroduction of lactose to tolerance, it's important to only increase by 1 gram per day and adjust the dose gradually over time. What's important is that calcium is still well absorbed from milk despite people having lactose intolerance. Sources of dietary lactose would include the naturally occurring sort of milk products that contain lactose naturally, but also lactose is added to a variety of different commercially available foods, and it might be used as a bulking agent. It might even be used as a filler in pharmaceuticals, and it's useful in baked products as well because it helps to increase the browning of them. The average intake of lactose in Sweden is around 12 grams per day and is reported to be around 7 grams in IBS patients in the UK. In terms of lactose sources in dairy products, you can see that milk has a high lactose content of 15 grams per 250 mils, whereas other products are much less than that. So yogurt is about half of that at 200 grams, 8 grams. And what's important to note here is that a lot of lactose gets converted to lactic acid during the maturation process of different dairy products. So for example, cheeses like ricotta and cottage cheese have a moderate level of lactose at 4 grams per 3 tablespoons, whereas cheeses that are very mature, things like cheddar, brie, Stilton, have a much lower level of lactose. And I think it's really important to note this and to emphasize to patients that this is key in their management. What about the different types of dairy milk that you might have? So semi-skim milk is usually around 4.7 grams per 100 grams. Whole milk, similar, 4.6 grams. If you were thinking about the condensed milks that you get in tins, this is 12.3 grams, so much more concentrated. And dried milk, dried skim milk, is 52.9 grams. So lots of products where there's dried skim milk added, it's a very high lactose source. Evaporated whole milk is 8.5 grams. Goat's milk is slightly less than cow's milk at 4.4. Human milk is more at 7.2 grams and sheep at 5.1. Suitable foods for lactose intolerance. So things like milk and milk products. Usually I would say that people can tolerate 4 grams of lactose at each sitting without any signs of symptoms. However, this is very much on a personal subjective level, and some people find that they do get symptoms even with having a small amount of lactose in their diet. Lactose-free milks, plant-based milks are a good alternative, so things like soya milk, rice milk, oat milk, nut milks like almond or hazelnut or macadamia. Hemp seed and coconut and quinoa are also used as plant-based alternatives to milk. What's an important thing to consider with the plant-based milks is that they need to be supplemented with calcium. We'll talk about that a little bit more in a moment. So lactose tolerance, as I mentioned, there's an individual perception of symptoms, and it's important to discuss this with patients in terms of their own personal plan, in terms of whether they want to try and include some lactose containing foods in their diet. So up to sort of 250 mils of milk spread across the day is generally quite well tolerated, and this may be an important consideration if we're thinking about the availability of milk and dairy foods across the globe, and how well these things are tolerated as part of a diet, of a sort of a healthy balanced diet. And it does depend on lactase activity, which has already been discussed. What's important to think about is the impact of transit time as well. So if you're having dairy foods in isolation on their own, you may find that the transit time is being increased. Whereas if they're part of a mixed meal, you might find that that actually helps to improve the tolerance of the lactose in that meal and the ability for the residual lactase to still absorb, break down the lactose so that it's absorbed in the small intestine. There's no evidence that with lactose you can have any sort of long-term small intestinal adaptation. Maybe probiotic and lactose as a prebiotic is something that I'll discuss in just a moment. And this is particularly in the case where lactose gets fermented by the colonic bacteria, and this helps to improve the tolerance of lactose in the diet. Other things to consider would be that mixed meals can improve tolerance. Cereals in particular can increase the energy content and delay gastric emptying, so it helps to improve the speed at which the lactose is passing through the gut, and so helps to improve lactose breakdown and absorption of glucose and galactose. Supplemental lactase is not always effective. So this is where you might be giving, someone might decide to take some lactase enzyme. Sometimes it might be helpful. It might be helpful on occasion if they're eating out. And the evidence is variable really in terms of whether it's particularly effective or not. Food temperature can also affect transit time. So very hot and very cold foods might be less well tolerated. So it's important to think about that in terms of delivery of lactose into the gut as well. Other considerations. So thinking about whether the diet can be nutritionally complete without lactose, it's important to remember that dairy foods contain calcium and vitamin D, which are particularly important for bone mineral density, and dairy-free alternatives may be supplemented. They may be more expensive and the nutrients may be less bioavailable, so it's important to consider these and to really think about whether plant-based alternatives are the right approach for patients, or whether you should be really thinking about trying to improve their ability to have a sort of an acceptable level of lactose containing foods in the diet to help with the absorption of calcium and vitamin D in the diet. It's important that we reintroduce lactose to tolerance, but also always we need to discuss that compliance and acceptability with people to see whether or not it's something that they really want to try and do. And if they don't, to really support them with avoiding those dairy foods and making sure that their diet is as complete as possible. Many people with lactose intolerance don't actually have lactose malabsorption. And are the symptoms due to something else? Are they due to other fermentable carbohydrates? There is an overlap of lactose intolerance with irritable bowel syndrome, and is milk avoidance really necessary? Let's sort of think about that prebiotic effect in just a moment. So just recapping on the symptoms again, and the fact that this overlaps with irritable bowel syndrome. In terms of that diagnostic criteria for irritable bowel syndrome, the row 4 criteria is what we use these days looking at irritable bowel syndrome. I'm not going to go into lots of detail about that today, however. There are guidelines for the dietary management of irritable bowel syndrome, so please do have a look at those. And the milk and lactose tolerance does come up as an issue on its own in isolation, but also as part of the low FODMAP diet. I mentioned that I was going to talk about the prebiotic effects of lactose, and this paper by Jan Geertz came out a few years ago now, looking at people with lactase non-persistence and giving them a repeated increased dose of dietary lactose from 3, 6 to 12 grams twice daily over a 4-week period, increasing gradually. And what the study showed was that it increased the gut bifidobacteria. It also reduced hydrogen production through breath testing and symptoms of lactose intolerance. So it's really important to remember that people with lactase non-persistence can tolerate lactose, and this study showed that up to 24 grams of lactose per day was well tolerated. So just to conclude, diagnosis of lactose malabsorption and lactose intolerance is challenging. It's really important clinically, and it's important to only reduce dairy products and to not exclude them unless it's something that someone really, really wants to do. But most people will tolerate some lactose. Be aware of dietary calcium and vitamin D needs, and remember that mixed meals, gastric emptying, transit time will improve tolerance. Lactose intolerance may also be associated with irritable bowel syndrome, and other dietary restrictions need to be considered. And it's important to consider the prebiotic effects, and gradual reintroduction can help that colonic adaptation. Thank you very much for listening.
Thank you. And that was a lot of practical guidance on the inclusion of lactose in the diet based on the various facets of lactose intolerance, which may be there in individuals. And what was also interesting was to understand about the prebiotic effects of lactose. We'll now move on to the panel discussion. And we have 3 panelists here, Professor Corinna Walsh, Professor Michael Gansel, and Assistant Professor Andrew Szilagyi. And we've already introduced the panelists earlier, so we'll directly go on to the questions. And some questions have already been answered on the Q&A column by Corinna and Michael. I'm really grateful for that. And we'll just take on a few. Oh, in fact, when I opened it a few seconds back, there were several questions, but there are some additional questions which I would like to ask the panelists. And there is one question which has just been posted. What is the difference between lactose intolerance and IBS? And are the nutritional managements the same? Corinna, would you like to address that, or anybody else?
I can speak to that, or at least start. The overlap of the irritable bowel syndrome and oligosaccharide intolerance is very high, but that is not specific for lactose. It also individuals who are fructose malabsorbers often report irritable bowel syndrome. And again, in a Canadian context, those with sucrose intolerance are also at a much higher risk of irritable bowel syndrome. So this is generally related to non-digestible sugars or oligosaccharides and not specific for lactose.
That's true.
Okay, thank you very much. Another question which has been discussed and thought of many times is, should nutrition recommendations differ more significantly between populations with varying levels of lactase persistence? And what challenges would exist when we are looking at these varying levels with nutritional adequacy, particularly in the vulnerable groups, and when we are avoiding dairy also? Again, it's open to any of the panelists who would like to take it up.
I wouldn't modify directions, particularly for people with lactase non-persistent, because there's, I think, relatively enough evidence to show that intolerance symptoms and IBS tend to be somewhat evenly distributed between both genotypes and malabsorbers. However, as I said, there's increased sensitivity in IBS, and as was said, it's non-specific. But recommendation differently may not be worthwhile, especially since we do not know whether adaptation in the LNP populations may be a way of improving or preventing several non-communicable diseases.
Thank you. Thank you. There is another question which has been just posted, which says,
I can maybe add
Sorry,
one aspect to Andrew's answer. Part of the treatment of lactose intolerance relates to simply taking away the negative perceptions of lactose in dairy products.
Yes, absolutely.
In many of those perceived and self-diagnosed food intolerances, and I know that better for non-celiac wheat sensitivity than for lactose.
Yes.
You have a nocebo effect, which means the symptoms are there only because the consumer expects symptoms. And I think taking the negative perception away will help to encourage people to drink as much as they can tolerate.
You know, lactose has a nocebo effect attached to it. So that goes along with what Dr. Gensel said.
Well said. Well, I think this question has already been answered in the presentations, but since somebody has raised it, we could just maybe reinforce it. Why do some people get issues of lactose intolerance with liquid milk, but less when milk products are consumed?
Other milk products. Okay, so I think you're quite right that we have spoken about this in the presentations, but I think it's because of the lactose content of fluid milk that is much higher than in many of the other dairy products. So in our fermented dairy products, the lactose content is far lower, and then obviously the effects will be less.
Yeah. Okay, there is another question which is very specific to diseases. So I'll read it, and then anybody who's willing to answer that. Lots of patients with myasthenia gravis that are on mestinon develop lactose intolerance. Is there a relationship or scientific evidence of lactose intolerance and myasthenia gravis as an autoimmune disorder? Would anybody like to talk about this? Very specialized.
Okay. I don't know specifically about myasthenia gravis. However, there is a lot of interaction between gut bacteria, perception of symptoms, and there are now a number of situations where the enzyme lactase can be modified by some processes. Like when I was looking up genetics of lactase, I came up on a transcription factor, PDX, which seems to be stimulated by, quote, iron deficiency, which is not, necessarily associated with myasthenia, but just to give a general example, PDX downgrades lactase, so inhibits. And then also that conference, there was a discussion about rifaximin, an antibiotic raising intestinal lactase. Thyroid hormones, insulin, corticosteroids may have an effect on lactase enzyme also. So I can't give a specific example in this case, but there are situations where lactose intolerance becomes an issue. For example, when about 25, how I became interested in lactose per se was through a clinic evaluating a African American male who was like in his 30s, was able to drink milk and no problem whatsoever. However, in the last several months, he noted lactose intolerance. Turned out that he developed thyrotoxicosis, hyperthyroid. And so whether the disease affects transit, gastric emptying, or intestinal, or a direct effect, have to be looked at specifically at the disease, but it is possible.
Okay, thank you very much. What are the most important unanswered research questions or gaps you think in dairy nutrition or lactose research? Maybe I request each of you to come on this very briefly. Your ideas. So Corinna, would you like to go first?
It's an interesting one. I think there's a lot of research opportunities when it comes to the field. So one of them would be, for example, looking at whether normal or usual dairy products like full milk with lactose has got different effects on outcomes compared to, say, milk where the lactose has been added and we've got the galactose and the glucose. So I think there are many questions about whether that affects satiety. We know it could influence the glycemic index of the product, but I think there's a lot of opportunities and a lot of studies that we need to do to be able to see, for example, with calcium absorption, is it the same in people using regular milk compared to milk where the lactose has been broken down? And we do know that it does affect the microbiome differently. And I think we've spoken about that quite a lot. But that's something that I think is a research opportunity. Thanks, Seema.
Thank you, Michael.
The one question, and that occurred to me when I was doing the presentation, is how fermented dairy products with mesophilic cultures impact lactose digestion. And the difference is that yogurt starter cultures metabolize metabolize lactose with a beta-galactosidase, which means if the cell is dead, the enzyme remains active. But most of the mesophilic starter cultures metabolize lactose with a phospho-beta-galactosidase, which means the enzyme is inactive if there isn't a live cell with active metabolism. And it occurred to me that there is good documentation on yogurt, but very little documentation on all the cream cheeses and buttermilks of this world, which are fermented with mesophilic cultures. So that would be a question which is in the back of my mind. What happens with these products if they're ingested?
Thank you. And Professor Zelazny?
Well, my main question, there are two actually. As I said, I sort of focus on the effects of dairy products and lactose on diseases. So in that context, one of the problems with looking at dairy foods and effects is what specific dairy foods should we be studying? For example, is it total dairy foods, milk only? Is it a nutrient? It was mentioned that we shouldn't use that. I believe the Thorning hypothesis is a reasonably good one, and maybe that's what one should concentrate on. But an important issue, which I try to point out, is that if it's true, and it was a hypothesis, although there's limited data, that there is a group of lactose digesters who are actually genetically LNP, but they are hidden in a group of studies where you're looking at dairy foods on disease effect, and a part of that population in mixed cultures particularly would be consuming dairy, but they would be unrecognizable without a biomarker. And so one of the obvious questions is, if that's true, do they have an impact on the outcome, perhaps through a Simpson's paradox effect? The reason I say that is we did a study a long time ago where, and it's the only study that I know of that exists, where we divided a meta-analysis of the effects of dairy foods on colorectal cancer outcome, 80 studies, and divided the world into relative homogeneous LP, relative homogeneous LNP, and mixed populations where the LP-LNP fraction was 4 to 6. And the two homozygous groups were significant, albeit modest, but the mixed group was numerically lower, but not significant. Took me 20 years to think of Simpson's paradox as a possible other explanation besides other confounding problem, but that study should be repeated and see if it's true. And then we can look at perhaps the better how dairy foods affect disease outcome. Thank you.
Thank you. And I think the last question and a comment from all of you very briefly would be that today it's in fashion to avoid dairy. And how can we as healthcare professionals respond to the growing popularity of dairy-free diets, even among people without diagnosed intolerance? And what practical advice would you give to the health professionals who are supporting people to unnecessarily avoid dairy? So again, briefly.
Okay, I think it's very important to explain to people that avoiding dairy could reduce their intake of essential nutrients. So I think we need to focus on not only one aspect of dairy, but dairy as a whole food. So, I mean, we need to educate healthcare professionals and consumers about that. And then I think highlighting the impact of the dairy matrix is a critical aspect. And to explain that the components or the nutrients and the structure of food do not have individual effects, but that the dairy matrix has got unique health benefits. And I think highlighting that will hopefully, if people understand that, make them more likely to understand how important it is that they should be consuming dairy and that avoiding it is unnecessary in most cases.
I would
Yes.
I would fully agree. And in the last 6 months, I completed a book chapter on the sustainability and health benefits of plant-based alternatives to animal products. And the learning was twofold. First, the difference in sustainability between the soy and almond milks and dairy is not all that big, and probably completely disappears if you produce milk on grasslands which are not suitable for crop agriculture. And the second learning was the health benefits of low-fat fermented dairy products, including yogurt, kefir. They are so well documented, and that's not only attributable to the microorganisms, that's microbes times matrix with all the calcium and the protein. So until further notice, switching out dairy products for plant-based is not necessarily a health beneficial choice.
You know, there is so much talk about using pre-probiotics, and it's $1 billion industry. Milk is cheaper, or dairy products are a lot cheaper to achieve perhaps a similar benefit. And I think that should be emphasized to individual people or whatever, you know, anybody asking.
I
agree with everything what you said.
Thank you very much. That was a very insightful panel discussion, and I hope our audience is satisfied with the answers that they have received. So just to summarize today's session, we do know that lactose has several metabolic roles to play, and it's not only a source of energy, but has much more to do in the body. It's very important to differentiate between lactose intolerance and lactase non-persistence, specifically in adults.
Yeah.
And with the exception of milk, most dairy products are tolerated independent of lactase persistence. So the notion that lactose has to be avoided is misdirected, and we need a deeper understanding into this. And many people with lactose intolerance do not have lactose malabsorption. The symptoms may be due to something else. Of course, we have many, many more important messages which came in, but we have to move on. And we have, again, the two quick interactive questions which have come up, which we are going to again answer. And the first one is lactose can play a functional role in health beyond simply providing energy. I request the audience to give their options so that we can see what the results are. Okay, so there has been an improvement. The second question is, generalized recommendations to avoid lactose are scientifically justified for most adults. Again, you please give your vote. Thank you. Okay, could we see the results for this? Okay, so the number of disagree respondents have increased, again showing that our seminar has had a benefit. And then, of course, we'll come down to the next reminder again. Please remember that you may request for a certificate of attendance or a CPD certificate, and all relevant information will be shared in a follow-up mail after the event. We would also kindly ask all attendees to take a few moments to complete the IDF post-event survey using the link shared in the chat. Your feedback is very important to us to help us improve future editions and identify topics of interest for upcoming events. Many thanks for attending this IDF event. Goodbye, good night, good afternoon, whichever part of the world we are in. And thank you so much for joining us.